Let's use Quilt to design a CRISPR screen against enhancers. And to do this, we'll start with a scenario where we just ran ChIP-seq for NANOG in human embryonic stem cells. This is a transcription factor that's key for pluripotency, and when we do that ChIP-seq, we're finding over 13,000 ChIP-seq peaks in this case, if you're looking at these in something like a UCSC genome browser. And our question is, which of these peaks, which of these NANOG binding sites are functionally important for embryonic stem cells? And the first step for that we're going to do on Quilt is find out which of these binding sites has a functional enhancer region. So we'll go to Quilt, and we will go to My Datasets, where now you can just drag and drop files in. And so I'm taking the ChIP-seq peaks from this experiment, and this is actually from the UCSC genome browser ENCODE project, where I've been able to download these peaks, these ChIP-seq peaks, in just a tab delimited table where it's a .bed format. And what I can do is just take this file and just drag and drop it right in. So now we have these NANOG ChIP-seq peaks from embryonic stem cells loaded up into Quilt, and you're seeing this loaded, and it will finish up. There's just over 13,000 here. And the first step will be to find out where these peaks intersect with enhancers. So to find those enhancers, we're going to use kind of a tried and true method here for finding these, and we're going to use ChIP-seq from histone marks, where we are taking two activating chromatin marks and then subtracting a promoter chromatin mark in order to find those. And that's enabled by a Quilt functionality where we can do genome intersection and subtraction. But the first step is to find out which data sets we're going to need to do that, and we'll go down and look for the publicly available data sets, because a lot of the ENCODE data is in here, and we'll find human embryonic stem cells, or HESCs here. Click on that, and that tag, that kind of hashtag, will bring up all of the ENCODE data that has been tagged for embryonic stem cells. And what we're seeing here is that you have these marks like I'm talking about, H3K4 monomethyl, trimethyl, and then K27 acetylation. So if you click on these, you can visualize this data here, and what we want to do is make a local copy for ourselves to work on. So we can click Fork here, and Fork will bring this data then into our repository. So if we go back to datasets, here it is right here. Let's click in this data that we've forked, and what we want to do is quilt this data. And in this case, instead of matching on a given field within here, we're going to use genome tools, and we're going to use the intersect operator. And here's our data, and for any sort of genomic intersect like this, you need to specify chromosome start and end. This has already been done automatically by Quilt. And then for that right data, I've already forked out the data that we need for doing this intersect, where we're going to find that first set of the intersect between K4 monomethyl and K27 acetylation, and you can just hit intersect. This is then going to generate a new data set within Quilt. And you can see in the description here that it's auto-filled in, that it has kind of the history of how this is generated, where you have one data set intersected with the other, and it's going to be performing that calculation in the background and creating this, and now it's done. You can see that from that intersect we have about a little over 100,000 genomic intervals there. And then from here right away we can go back into Quilt and genome tools, and instead of doing intersect we can do subtract. Subtract then in this case operates the exact same way, except this time we are going to subtract out the peaks that would be on the promoter in this case, where it's K4 trimethylation. So instead of doing all this, we're going to do kind of a cooking show style, where I'm going to go back and skip right to the meat of things, where we want to take our enhancer peaks here and then intersect them with what I have identified here as the human embryonic stem cell enhancer. So I've renamed this data, whereas before it was kind of a long string of the history of it. I've just renamed this in the description and in the title, and you can intersect here, and that's where we're going to find enhancers that are bound by NANOG. So we've done the analysis to find the enhancers. There's just over 3,000 of the enhancers that are bound by NANOG, and then for a functional experiment, what we want to do is design CRISPRs to those regions. So to do that, we are going to go back to our data sets, and in this case we can look for the tag for CRISPR, and a lot of genome-wide libraries and also customized designs are in here. So one of these data sets is a set of CRISPRs that have been designed to all DNase hypersensitive sites in the human genome that fall over ENCODE DHS regions. So this can be loaded up in this case directly from the public data, and it can be forked over, and by intersecting that with the data set that we just generated with the NANOG bound enhancers, what you'll eventually end up with, and again, this is kind of like a cooking show where it's already going to be done, so we don't have to sit here and wait for all of it to finish. What you can see is that there are now CRISPRs that are designed to these over 3,000 different regions, where the CRISPRs here have already been designed. These have been filtered to not have off-target effects. And then here are oligos derived from this guide RNA 20-mer for either cloning these in sort of ordering them as a plate or ordering them as a chip-based oligo library. So from there, you can go back to the first video in this video set about designing CRISPRs to see how those can be cloned, and you can start your experiment. So this has been a quick video to see how you would do that for enhancers, not just for genes. I hope it's been useful. Leave comments if you have any questions, and good luck.